right now?
Yes.
Okay. So hello everybody. I'm Pratik Gupta from University of Minnesota, and I'll be presenting the CAMPI 3 bioinformatics study on behalf of the CAMPI 3 team. The CAMPI 3 study was aimed to evaluate bioinformatic workflows and impact of key decisions such as database selection, search parameters, and filtering strategies on taxonomic and functional annotations. The goal is to provide best practices for metaproteomics analysis. And to do this, we provided the community with two types of data set. One was a synthetic microbial community with known composition and a complex biological sample. The synthetic data set included four microbial mixes, each designed to test resolution under different conditions. The MIXA, for example, shown here, contained 16 bacterial species, and then some of these species, and all of them in equal amounts. The two taxonomically related species could be easily resolved. There were two impossible to resolve, and then there were three difficult to resolve. And we collectively call these collisional pairs. Whereas mix C had one of the species missing, and mix D also had another species missing. There was also a mix B sample which had a similar number of species, but they were in unequal amounts. The participants were sent mass spectrometry datasets along with protein sequence databases and were asked to report their search results using submission templates. And the study drew something like 21 submissions from 20 labs across multiple countries. And each of this workflow, the bioinformatics workflow that was used, was reviewed for the choices that affected the results. The outcomes revealed variation in taxonomic and functional annotations. So with this, you know, Jelio Alves from NIH, he assessed the differences in peptide detection using Wasser's time distance analysis, and this was used against expected biomass. In this analysis, normalized relative abundance values were used to calculate the biomasses. Each point that's shown here in this figure represents a sample submission. This plot also provides detailed view of the scatter plots that separate into two groups. So there is the group that is kind of away from the ground truth, which has been shown here in the left bottom corner, and some of them which are closer to the ground truth. So basically, samples that are closer to the ground truth show higher accuracy in peptide detection. Now, based on this analysis, Ben Kunat from the University of Luxembourg, he performed independent component analysis, or ICA, to go deeper into the data sets and dissect participant contributions. For this, he used abundance per taxon across samples. Later, he used a normalized metric contribution per participant to determine key performance metrics such as precision, accuracy, and overall score. The ICA process basically enables to identify key influencing features and link them back to the metadata and pipeline choices. Based on this analysis, Ben could rank these features into statistically significant features such as taxonomic resolution and annotation and others that have been shown in various shades of blue here. Examining features that distinguish these participant submissions for taxonomic resolution, he could cluster this across ICA components. The scatter plot here basically shows samples with similar signatures grouping together, and eventually we plan to look at the other significant features as well. We think that this analysis will help in recommending researchers on best practices for their own study and also for global study that was described earlier. Next, Lucia Drenga from French Alternative Energies and Atomic Energy Commission looked at peptide level quantitation. And for this, synthetic data sets that I had described earlier were used for this analysis. Lucia basically used three of these data sets since they had peptide intensities that were reported. Now, the mix A sample, as I mentioned earlier, has all organisms in equal amounts, while mix B has samples that are in different proportions. Now, when she measured the peptide ratios between these samples, you can see they fall below the expected values shown by the dashed line, and thus, basically, this shows a dynamic range separation in these data sets. I think we will be considering this in the new CAMPI quantitation study that we described and discussed later. The mixes also have three collisional pairs. And when Lucia looked at these collisional pairs, the volume plots showed that collisional organisms consistently show higher error as compared to non-collisional organisms, underscoring the impact on quantification accuracy. So in summary, basically these three bullet points just summarize what I described earlier, but to add to this, biological data set will be analyzed for factors driving participant variability. And then with this analysis, we will recommend bioinformatic best practices for the global study. And last but not least, this is the CAMPI3 team. We have been working on this for 2 years. And we hope that the study will be published early next year. And I appreciate your time for your attention. Thank you.